So I said at the very beginning here, plasma membranes can choose who comes across. When we say they can choose who comes across, we talk about as being selectively permeable. It allows some substances to come across, and it prevents other ones from coming across, okay? So it allows some and inhibits some others, okay? Which ones? Some, it inhibits polar molecules, so those that have a charge, those that aren't lipid soluble, those that aren't lipid soluble, big things, okay? Charged. Polar. Big. No crossy cross. Okay. If they're charged or have polarity, or they're they're water, okay, which has some polarity to it, those are always big things, okay, especially if they're water-based. They have a very hard time coming across, okay. If they're big items, they could destroy the plasma membrane, so they generally don't come across. So they're selectively permeable. I can bring those suckers across, but only if I decide I need them. Okay, so they're going to need a carrier protein that we talked about at the beginning with the protein's functions. Okay, so what kind of stuff comes across? Well, carbon dioxide, CO2, oxygen, O2. Okay, these things are non-charged. Right? So they could easily come across, which is fantastic, which is fan-freaking-tastic, because we're going to see these in photosynthesis. We're going to see these in cellular respiration. Okay, I need CO2 to be freely let go. It's a waste product for us. I need oxygen to be able to easily come in to make ATP. All right, so those two things are pretty obvious in that they can easily come and go as they please, especially as we start to look at chapter 6, 7, and 8. Okay. Glycerol and alcohol, they can also come across. They're very, they're lipid soluble, so we can bring those across, relatively easily in that regard. All right. But carbon dioxide and oxygen, I always highlight and underline those because we're going to see those a lot, and those are the best two to remember. Wink, wink. Another thing that we have to remember too when it comes to this, remember this phospholipid bilayer is a boundary, okay? Okay, so it keeps whatever's out, out. Okay. And it keeps mostly whatever is in. Okay. When it does this, guys, it creates this thing called a concentration gradient. Okay. It creates this thing called a concentration gradient. Does everybody see that there's more blue on the outside? Out, in. There's more blue on the outside than there is on the inside. So we've established a concentration gradient. Conversely, we can see that there's more green on the inside than there is on the outside. Again, we've established a concentration gradient. All right. This is what plasma membrane, cell membrane, whatever you want to call it, okay, helps us to create. It creates this natural gradient that occurs in every single cell. Okay. Establishing this concentration gradient ensures that I have high on one side and low on the other side. Okay. This is going to create this gradient that will allow the cell to bring across what it wants easily without energy, okay? Going from a high to low concentration. It is this establishment of a concentration gradient that allows neurons to rest, a muscle to relax or contract, for smooth muscle to contract and move food forward through the digestive tract, okay? To be able to form urine, a concentrated urine or a dilute urine. So this concept of this concentration gradient that is established by this plasma membrane is an important concept, and it's going to drive the next several slides. Okay. I don't know why. I meant to move this. I don't like how they put this here, but okay. Water can move across the plasma membrane, but we need specific carriers. You know, aquaporins. These are protein channels that are specific for water only. We have a number of them scattered throughout the body. Usually in the kidneys is where you're going to see them, majoritively. But they're going to help bring water across into a cell because, you know, cells are going to be able to have water and move in and out. So we have these things called aquaporins, and we have hormones, right? Antidiuretic hormone, vasopressin. We have a number of different hormones that will, you know, put these specific protein channels into cells so we can absorb more water, reabsorb more water, or excrete more water, okay? And this is what helps us to drive this ability to form a lot of pee, a lot of urine, okay? So water can cross the plasma membrane. It has a specific name that we'll talk about here in a second, but it's basically done through this thing called aquaporins, a specific protein channel. How would you see this on the exam? Probably as a multiple choice type question, okay? We can move ions, charged ions, polar ions across with assistance, okay? Where we have these carrier proteins. We'll talk about that here in a second. These are all going down a concentration gradient, okay? There's going from a lot of water and bringing water in, bringing stuff across the carrier, bringing stuff across the membrane with a carrier protein, coming from a lot to a little, from high concentration to low concentration. Sometimes I got to go backwards. Sometimes I got to pump stuff out that's low and I got to pump it into the high. When I go against that concentration gradient, that's active. And that costs money. Okay. And what's money? ATP. Okay. We call this bulk transport. And we're going to talk about exoendocytosis, so I don't know really why in the hell they put it there. All right. So we're going to pretend that it doesn't exist, and we're going to talk about it here in a second. So again, here's my barrier. Okay. Create a concentration gradient. Charged stuff is repelled. Charged stuff is repelled, okay? Non-charged can easily go through. Large things are repelled, okay? And then I have my little specific carrier protein, my aquaporin, to help bring specifically water in. I'd put 1,000 stars on there, but I don't want to do that. These 4 things you need to be able to describe. These things you need to be able to talk about. Tell me how the process works, give me an example, all that fun stuff, okay? This is obviously a fantastic short answer question, okay? How many are going to be on the exam? I don't know, okay? I try to do 1 short answer exam question for each chapter, but we'll see, okay? But obviously this would make a good short answer question. You have seen the exam, you probably know. I mean, I tell you, I'll give you 2 or 3 examples of great short answer questions for each note set, okay? You should be able to discuss those, okay? When I ask you to list, just list it out. When I ask you to describe the list, describe it. Just write down, you know, what are the 4 methods for movement of molecules in and out of a cell? You just would write down diffusion, facilitated diffusion, osmosis, active transport, bulk transport. If that's all you listed, then you're only getting about a point, okay? You got to explain if I ask you to explain. Some of you did not do that on this exam. All right, so let's talk about these. The first thing we need to establish, though, I need to establish some words, okay? So let me look at these words here, okay? A solution. Here's my beaker. Inside my beaker is a solvent. That's the liquid. Okay, that is my solvent. The solvent dissolves stuff. Solute is what I dissolve in the liquid. Okay, it's the red. So solute is red. And the solvent is blue. Okay, because we're going to be talking about stuff going in and out of a solution. We're going to be talking about the solutes and how that changes concentrations of fluids. Okay, solutes dissolve. Solvent is what dissolves those substances or tries to dissolve those substances. Okay, so that's the basic terminology there that we're going to be talking about everything here. Okay, a solute, substances. Solvent is the liquid that we dump them into. Okay, so I'm going to have diffusion, osmosis, and facilitated diffusion. Okay, all of these types of transport mechanisms are all passive. Okay, are all passive. Okay, passive means you just sit back and it just happens. You don't have to expend any energy to do it. Okay, so for passive, no ATP is spent. Okay, the first passive mechanism we have is right here on this slide, and this is movement of molecules down a concentration gradient. What in the hell does that mean? From high to low. From high to low concentration. In brackets is just shorthand for concentration. Okay. So diffusion, net movement of molecules. Net movement of molecules down a concentration gradient from high concentration to low concentration. All right. Movement occurs until everything is equal. Okay. Movement occurs until equilibrium is met. Okay. And that's what they're trying to show you here. They're dropping a little crystal dye into this fishbowl here. And here you can see, here's the dye. Okay. Nice, solid, rigid structure. As it starts to dissolve, okay, you can see all these little dye molecules start to spread out. Okay. They were just here. Okay. Now they're spreading out. And they're going to continue to spread out until they've reached equilibrium. And that's what we see right here. When this fishbowl has turned this beautiful shade of purple, that purple dye has distributed equally all throughout. Now those dye molecules just kind of hang out. They don't move around anymore. Okay. they have reached equilibrium. So for diffusion, this movement of molecules occurs until that concentration is uniform. They're equally distributed. That's all that means. There are a number of things can affect this, and when you do the lab for cell membrane and transport, I imagine they'll hit on this. I can't remember exactly. I did all of these a month ago, so I can't remember all of them off the top of my head. But there are a lot of things that can affect these things, guys. No problem. Temperature, pressure, electrical currents, molecular size. There's a lot of things that can affect this. Temperature is probably the most obvious. If I was to heat that solution there with the dye in it, think of it in terms of you. If somebody put you on a big frying pan, okay, that was cold, or let's just say room temperature, you'd put your bare feet right on it, right? And you'd sit there and stand. You'd be like, this isn't too bad at all. I'm just going to stand right here. You maybe lift up your foot here and there. No big deal. You're walking around. But if all of a sudden, someone turned that pan on and you're standing on it with your bare feet, all of a sudden you're going to start moving around really fast. Try to get off that sucker. That's what temperature does to these molecules. It speeds them up. They start bumping into each other. Okay? They start having more interactions and reactions, and that's going to help speed up that spreading of that dye around. Pressure does the same thing. It's just kind of hard to visualize, but think of if all of a sudden I'm this. Okay, obviously there's a lot more pressure. I have a smaller space, so stuff's going to be hitting and bouncing a lot more. So that's going to speed up the rate of that dye spreading out. Same thing for an electrical current, same thing for molecular size. The bigger the size, the faster they're going to move because they're bumping into things and they're going to move around a hell of a lot faster. All right. So there are a number of things that can affect this. And we may hint on some of these later when we talk about some functions later on down the road. Okay. Here is a great example of diffusion. Okay, this is an example. This is diffusion. Okay, here's diffusion. I have a high concentration on one side of the plasma membrane, and I have a low concentration on the other side of that plasma membrane. Okay, so where is all this oxygen going to go? It's going to go here in the alveolus, going to go into that little blood vessel. Every time you take a breath in, you breathe in all those oxygen molecules. So now there's a high number here. That creates a huge concentration gradient. High here, low here. Okay? So it's driving all this oxygen into that little blood vessel. And that little blood vessel helps to distribute all that good oxygen throughout the body. Okay? So diffusion is something that's used quite a bit. It's used in the lungs, it's used in the capillary bed, used in the kidneys, used in a lot of places. My second type of passive transport is osmosis. Osmosis is basically diffusion for water. Okay. Okay. This is diffusion of water. Movement of water from high to low. Okay. Movement of water from high concentration to low concentration. All right. All right. So here, this is the concentration of the water, okay? Or the concentration of this solution here, okay? Solution consists of water and the solutes. There are a greater number of solutes here, okay, oops, 10% solutes, okay? And this, outside it, only has 5% solutes. Think in these terms, okay? Everything has to equal up to 100, right? So if I have 5% solute, I have 95% water. If I have 10% solute, I have 90% water, okay? I assume you're all with me on that basic math. 90 plus 10 is 100. 95 plus 5 is 100, okay? So we're all there. Which one has the more water? Outside has more water, right? It has 95 compared to 90. So water is going to go in the direction from high to low, okay? So water is going to move in this direction here, okay? So you can talk about osmosis in terms of water movement. Osmosis is movement of water from high concentration to low concentration. You can also talk about it in terms of solutes, okay? Water is going to go from where there is fewer solutes to where there is more solutes, okay? Because what does it want to do? Dan, there we go. Because if we move water across, what is that going to do to this solute concentration? What do you think it's going to do? If I have more water to this, is this going to dilute it or concentrate it? Who wants to unmute and take a chance?
I think it's going to concentrate it.
If the water moves in, okay? If the water moves in, if water is going from high to low and more water comes in, now I'm adding more water to this solution here. So that's going to dilute that solute concentration. And as the water moves across, it's going to create a much more equality between those percentage of solutes, okay? So this is going to get a little bit more concentrated, and inside this little tube here is going to get a little bit more diluted. Just think of what happens when you add, coffee, okay? You've poured your cup of coffee in the morning from your little coffee machine, all right? And you've added some sugar to it, okay? You take a drink, you're like, oh my God, I've added too much sugar. How do you make that sugar less concentrated? Do you get a spoon and spoon out the sugar? No, it's dissolved in there, right? All the little molecules are in there already bouncing around. So how do we dilute that coffee so it's not so sugary? You just add more, right? You just add more, okay? Yeah, you add more coffee. Perfect, okay? So the same thing is happening here, okay? As water goes from high to low, okay, that's going to take less water. It's going to take away water from outside the solution here, outside in this beaker, and it's going to go inside this little thistle tube here. So inside becomes a little bit more diluted, and outside now becomes a little bit more concentrated. Ultimately, it wants to do what? Why is my thing not working? Ultimately, it wants to go until we have equality, okay? Everything's at equilibrium, right? Where's my, yep. What are we doing? What are we moving to? All these things move until we reach equilibrium, okay? That's the beauty behind passive transport mechanisms, all right? They go until they've reached equilibrium, whether it's diffusion, movement of molecules from an area of high concentration to low concentration, high to low, okay, as it spreads out. Or our example here, high to low, molecules move from high to low, or whether it's a specific type of osmosis, or I'm sorry, of diffusion, osmosis, movement of water across the selectively permeable membrane from high concentration to low concentration. Okay. This gives us what's called osmotic pressure, and we'll see that here in a second, okay? Because we're going to expound upon the movement of water and the concentration of what happens inside and outside of a cell. Here is a little video. I'm not going to click on it, but please watch it, okay? I have a few different videos embedded within here that go over osmosis and diffusion and facilitated diffusion and all the fun stuff that happens there. So please just watch these that help to reinforce what we're doing here in class. So go ahead and take a look at these, and hopefully maybe it'll make a little bit more sense than what I'm spouting off through my mouth. I don't really know. Sometimes I just don't say it too well. All right. So this ability for water to move in and out of a cell is actually a pretty crucial thing here. It can help change the concentration inside of a cell dramatically, okay? Just by giving somebody too much of a solution or too concentrated of a solution, like saline in a hospital setting, if we give them too high concentrated of a saline, that can really affect how our cells survive, okay? You see it in a plant as well, but it's not, there's not this huge kind of bursting effect and death that occurs. It's a little bit more different. But here we have three types of solutions, okay? You can have a solution. You can see a solution right here, okay? That is equal on both sides. So here is that's my cell, okay? Here's my cell. And here's my solution. Solution. Cell. Okay. In an isotonic, everything is equal. Okay. Everything is equal. So let me change my color here. Okay. Let's just say this is 90% water and 10% solutes. Okay. In here, 90% water and 10% solutes. Okay. So everything's equal. You have, let me change my color again. You have a little bit that goes out, but for everything that comes out, you have just as much coming in, okay? Because there's always movement across the plasma membrane. It's not static. It's not made of iron. It is a fluid mosaic model. So it has fluidity to it. So stuff is going to be constantly coming in and out, all right? So everything that goes out equally has to come in. It is iso. Iso means the same. Okay, ISO means the same. Change my pen color. I'm having too much fun now. Tonic is strength. Okay, so they are the same. Same on both sides. Same on the inside, same on the outside. All right, so that's isotonic. All right, now let's look at a solution that is hypotonic. Ugh. Okay, so here's my solution again. Okay. concentration of the solute in the solution is lower than inside the cell. Okay, so concentration of the solution is lower. Okay, so here we'll say this is 5% solute. And here we'll say this is 10% solute. Okay, so the concentration of the solute in the solution is lower than inside the cell. Okay, so would everybody agree? 5 is less than 10. High five, sweet, awesome. Okay, so where's water going to go? Okay, where's water going to go? Where's water going to go? Where's water going to go? Let's try here. Okay, so water is going to go from high to low concentration. So if this is 5% solute, it's 95% water, right? W-A-T-E-R. I'll just do that and do H2O because I think that's what I was going to do anyway, okay? If this is 10% solute, 100 minus 10 is 90. So that's 90% H2O, okay? So where's water moving here? From high to low. So water is going to go into the cell here. So water is going to go into the cell. Let's try yellow. So a cell placed in a hypotonic solution. I don't like that. It's going to swell. Sorry. Okay, I'll use a highlighter next time. All right, so cells, when they're dropped in a hypotonic solution, lower concentration of solute inside the solution than inside the cell, that's going to cause the cell to swell up. Okay, and that's what you see right here. More water's coming in than going out, and that causes this cell to blow up. And that's called lyse. In an animal cell, this is death. Okay? If that plasma membrane loses its integrity and bursts like that, all that stuff that's inside comes out. Well, now the body's like, hell, I got to take care of this now. So goes and kills that cell completely, and it's done. All right? So if you have a number of cells placed in this hypotonic solution, boy, that can cause a lot of cellular breakdown. You can destroy all those red blood cells. They start to clump because they've lysed. And now you get all this excess stuff in your blood system, and it tries to mount an immune response to clean it all up, and dead. Okay. So in animal cells, I'm going to change my color here while I'm thinking about it. Okay, so in an animal cell, it's going to cause that thing to lyse or to rupture, explode. All right. In a plant, it's freaking awesome. Okay, more water comes in, but what does what does a cell have? A cell, good God, what does a plant cell have compared to an animal cell? It has that cell wall. So it doesn't necessarily get to that point where it just expands, and blows up. It expands, and then it bumps into that cell wall, and it continues to exert force and exert pressure. This is called turgor pressure. It allows that plant cell to stand straight on up, erect. So when you water your plants and they get more water, as they bring more water in, that fills up that large central vacuole there, and it pushes up against the cell wall, and then that thing stands on up. Protozoans have these things too, but I don't really care about protozoans. I know I'm recording this, and I apologize to all the biologists in the department. Okay, there are some protozoans. This is how they get rid of excess water. Protozoans can go in and out of different environments that have a higher concentration. So, I mean, they're important. Okay, but I like to look at it from the aspect of a human being. Okay, But there are protozoans that live in freshwater environments that all of a sudden would come into a higher concentrated water for whatever reason. And so they have these contractile vacuoles that they'll pump all excess water into those things so they can become isoosmotic with the surrounding environment. So there are some animal creatures that have similar functions to a plant cell, but we're not protozoans. So I don't really, that part doesn't really, I'm not really concerned about that part. Okay. In the last solution, hypertonic, hyper means elevated, okay? Means elevated. Someone's hyperactive, right? They're all over the place. Elevated, okay? The solute is higher in the solution. Solute is higher in the solution than the outside the cell, okay? So here's my solution. And it has a higher concentration. So we'll say 10% solute. And 90% water. Compared to inside the cell. That is 5% solute. And 95% water, H2O, okay? So in this case here, guys, water goes from high to low, okay? So more water is going to come out. More water is going to come out, okay? That's gonna cause this animal cell to lose size, to shrink, okay? It's going to cause it to shrink. In an animal cell, we call this crenation. That's when it shrivels up. If I go here, you can see more water is leaving than coming in. So this thing shrivels up, and we call that thing crenation. Again, not a good situation to be in for us, animal cells. Plant cells undergo this process called plasmolysis, okay? Think of it this way, and they do a good job of kind of explaining it there, all right? But here, you know, think of this plant next to your driveway, okay? In the wintertime, to get rid of the ice, you throw a bunch of salt down. So the outside of the cell is going to have a much, much, much higher concentrated environment. Well, the plant, you know, has it, you know, and then as you scrape, you know, the snow and the ice off, you shovel it off into the grass right next to the thing, right next to the driveway. And now all of that fluid dissolves and all that salt gets into the surrounding dirt. And now the plant roots are absorbing all of that higher concentrated water. Well, when that happens, the higher concentration now, all the water that's inside this large central vacuole, okay, all of that's going to start to dump out, okay, because it's drawing the water out. So that plant cell that was standing up tall and erect, all of a sudden starts to shrink down, starts to shrivel, just like it did in an animal cell. But we have a plasma membrane here, okay? We have a cell wall here. So now I don't exhibit any more turgor pressure, and what happens to that plant sitting next to your driveway? It starts to wilt, okay? Why does it do that? Because we are drawing water out of that large central vacuole. If we remember way back to chapter 4, we talked about large central vacuole being this huge reservoir to just store excess junk in or water in. And that's what it helps to get plants to do. When the situation arises, it gives them a water source. Will they wilt a little bit? Yes. But will they die? Not necessarily, okay? Not necessarily. Okay? But that allows these plants to survive because I have all this water inside here. H2O. So the plant just draws upon that water. It's able to survive. Yes, it may, look like the dude from Weekend at Bernie's, but at least it's alive. Okay. Another video here for you to watch and enjoy. Okay. I know we're kind of getting late here, but getting past our lecture time, but I want to go over a few things here with you guys. Okay. So we'll continue on here. My last type of passive transport, because we've had diffusion, we have osmosis, and now we have facilitated diffusion. Okay, facilitated transport. I don't know why they call it transport. Okay, most textbooks refer to this as facilitated diffusion. Okay, whether we call it facilitated diffusion or facilitated transport, we are facilitating something. When you facilitate something, you help, right? When you facilitate, you help. Okay, when you facilitate, you help. So we are helping things get across this plasma membrane. They're still going down their concentration gradient. They're still going down from high concentration to low concentration. Again, this is passive, so still no energy required, okay? No energy required, all right? So facilitated transport is passive, even though I'm using a carrier now, okay? Combined with a specific carrier protein to move the substance across. Why? Because it's big, okay? Glucose is big, okay? Amino acids have charge to it, all right? Again, charged substances don't come across the plasma membrane. So amino acids, a lot of them will have a charge to them, all right? So I have to facilitate their movement across, all right? So I'm facilitating the movement of this across through this carrier protein. All right. So when I say facilitated diffusion or facilitated transport, it's still passive. So no energy, no ATP. And it's going down its concentration gradient, going from high concentration to low concentration. Easy. Okay. To go down the slide is very easy. All right. What uses this type of mechanism? Big things, charged things. Okay. And here you can see this is a big thing. Okay, these are big, big. Okay, so if it came across that plasma membrane, it would destroy the sucker. You don't want that. I have a specific carrier protein to bring this thing across. Okay, maintain the integrity of the cell. Still went down its concentration gradient from high to low, so no ATP required here. So facilitated transport. And that's what this video shows. Shows you some diffusion, shows you some facilitated transport. Fantastic. Please watch these videos. They're going to help maybe make it a little bit more palatable for you. Okay. The opposite of active, or I'm sorry, the opposite of passive is active. Whenever you see active, two things. Okay. It requires money. Okay. It requires ATP, okay? And you're going against the concentration gradient. You're going against the concentration gradient. With passive, with all of those things, guys, they went from high concentration to low concentration, right? So they're going down. Whenever we go down, we're going with the concentration gradient, going with, okay, going from high to low. With this one, we're going against it, so I got to go up. To go down the slide requires no input. To go up the slide requires some effort, all right? You're going against that concentration gradient. So you're going from high to low. Movement from movement from low to high. Movement from low to high, okay? You're going against it. There are carriers for this one, okay? Because to go against a concentration gradient just doesn't willy-nilly easily come through the plasma membrane. So we're going to need carriers for this one as well. So just like facilitated transport, active transport requires some carrier proteins as well. Okay? Because of that one simple fact, most people, a handful of people will get this wrong on the exam. Okay? And I don't want you to that. I don't want that to be you, but it's going to happen. So please do your best to try to remember facilitated transport, down the concentration gradient. No ATP. Carrier protein though. Active transport goes against the concentration gradient, goes from low to high. Okay? That requires input of energy. That requires a carrier protein as well. Okay? The classic example is the sodium-potassium pump. Where do you see that? Everywhere in the human body. Muscles, nerves, everywhere. Okay? And the video to prove I'm not a liar, but here's this video. I have a specific shape carrier protein, specific shape molecule, sodium. Okay, load them up. It requires energy. It changes shape. Oh my gosh, I get that sodium out of the cell. Okay, then to reestablish that, potassium, specific shape, potassium loads on up, allows me to attach that phosphate back to an ADP molecule, and I'm reset, and I'm ready to do this all again. We're going to see this sucker ad nauseum when we start talking about metabolism and cellular respiration. So you're going to see this again, okay? So understanding this is going to be an important kind of concept here. All right, so we had passive, diffusion, osmosis, facilitated diffusion. We have active transport, we just saw right here. But we have different types of active transport. Remember that slide that we talked about with bulk transport? Well, here it all is again. So I don't know why in the world they had that out there. It makes no sense, okay? But a specific way, a specific subset of this active transport is bulk transport, okay? Bulk transport is active transport. When we say bulk, we mean a large amount, okay? So whenever we want to move a large amount of fluid, whenever we want to move a large amount of solution, we term it bulk transport. It's basically active transport, okay? Because you're moving a lot of stuff at one time. If you wanted to bring in 25 bags of groceries, load them all up at once, it's gonna take a little bit more effort to move all that stuff, a little bit more energy, okay? As opposed to just bringing in one bag, okay? That doesn't take much effort, all right? So where do we see this bulk transport? We see it in the body, okay? We see this at the capillary level. All right, the exchange of oxygen and nutrients at the capillary bed inside organs and tissues. So we see this a lot within the human body. When we talk about this large amount of movement in and out of a cell, active transport or bulk transport, there are two major words that we describe here. Exocytosis and endocytosis. Cyte, osis of the, osis means of the cell, cyte means cell, exo of exo. Whenever you see exo, think exit, okay? Endo means within. Okay. So we have two ways to actively move stuff across, two ways to actively move a lot of stuff across the plasma membrane. You can either bring it into the cell, okay, through endocytosis, cells engulf and bring on in, okay, or we get rid of it, and that's exocytosis. With exocytosis, we are getting rid of. Okay, this is stuff that the cell doesn't want, that it's going to get rid of. So we have two types of active transport. Exocytosis removes it from the cell. Endocytosis brings it into the cell. To that end, we even have several different subsets of endocytosis, as you can see from the slide, obviously. Okay, these are all subcategories of endocytosis: phagocytosis, pinocytosis, and receptor-mediated endocytosis. Okay, a phagosome within the body. Phago means to consume, to chew. All right. So with phagocytosis, we are engulfing large things. Large things. It means cell eating, phagocytosis does. Okay. This is how white blood cells work in the body. All right. Lymphocytes, white blood cells, let's just say, they come up to a foreign invader. They look at his sugar coat. They're like, okay, this sucker ain't supposed to be here. And boom, okay, wraps around it, brings it in, and engulfs, and basically kills it in the process. Pinocytosis is cell drinking. Okay, this is where we bring in very small particles and some water. We used to just think it was water, but when they first coined this term, but better technology, better microscopes, they actually see that they're bringing in some very small substances across. So in pinocytosis, we are bringing in small substances and water. Receptor-mediated endocytosis, again, whenever we see receptor, guys, what did I say? Specific. Specific. Okay? Receptors are specific. We bring in a specific small substance and a little bit of water comes across as well. It's basically pinocytosis, just specific. Okay? I talked about this condition earlier, hypercholesterolemia. You're like, oh my God, that's a big word. Well, just break it down, okay? Hyper means elevated. Okay? Cholesterol, that's the word cholesterol, pretty easy. And emia means blood. Okay? So this is elevated cholesterol in the blood. There you go. Okay? Whenever you get lost in the biology realm, just break a word down into its component parts. Okay? That's why I keep dividing these words up for you. All right? That's how you figure out what in the hell they're talking about. Just break the word up. Okay? With receptor-mediated endocytosis, we have a specific receptor. Okay? With hypercholesterolemia, they have a genetic disorder where they don't have those specific receptors. Okay? And you can have the best diet in the world. You can exercise, clean diet, all that nonsense. But if you have that genetic defect, you're going to have cholesterol that builds up in your blood vessels, even though you may be spot on with your diet and exercise. Okay? So specific receptor binds to a specific substance, and that's what you see right here. Okay? Bringing in the specific substance, small, and some water. There's a phagocytosis action going on here. You can see the white blood cell extends out these little pseudopods that surround it and engulf and bring it in. Pinocytosis, small substances, and water. If I go back here, this is how we get rid of things. Okay. Here's exocytosis. So I've packaged up whatever I want to get rid of from the cell. You can see it comes up here and merges with the plasma membrane and ultimately dumps its contents out. They exit the cell. It's actually really complex, that interaction right there. I mean, this picture looks like, oh, hell, willy-nilly, let's go and attach. But there's actually a lot of stuff that goes on there. Okay? It's like a whole chapter in physiology that talks about just this one simple act. Science, man. It's crazy. Video for you to view and enjoy. Ah, crap. Wrong button. Sorry.